Good morning and good afternoon, everybody. Thank you again for taking the time out to register and sign up for our webinar and for attending. We look forward to talking to you today about our DiscoverX portfolio of assays that can enable the development of cancer immunotherapy drugs, all the way from discovery to combinations. My name is Avi Saharia, and I'm the director here of cell-based assays and biologics. And today, before we get started, I'd like to make a couple of administrative announcements on this webinar. This webinar is being recorded, and you will get a copy of the recorded webinar within the next couple of days after the end of this webinar. You will also get a copy of the slides for the webinar at the same time. If you do have any questions at any point during the webinar, please type them into your chat box or the Q&A box that you can find on the drop-down menu with the WebEx, and we'd be happy to address them at the end of the webinar. So without further ado, today I'm going to talk to you about the technologies that we've developed at DiscoverX that can enable your development of cancer immunotherapy drugs. I'm sure a number of you have already been quite familiar with this image that you see on the left side of your screen that highlights the interplay between cancer cells and the immune system. And this is dubbed the cancer immunity cycle by Drs. Daniel Chan and Ira Melman at Roche Genentech, summarizing decades of work through numerous scientists globally. Transitioning this body of work into a drug discovery process and then driving that to create safe and efficacious drugs to be utilized in humans is and remains quite challenging. And within that process of drug development, what we've done here is binned that entire process into 4 high-level categories, namely all the way from screening and optimization of lead molecules to testing for efficacy and biomarkers in a human tumor microenvironment and safety preclinical studies, and therefore, and even further downstream from that, looking at clinical combination strategies for your particular molecule. And the questions that often arise as you go through each of these stages are what we've highlighted below, and these are questions that we've often gotten from clients of ours coming into us and asking, for example, how do I screen for an active molecule and optimize for that using a cell-based assay? Or how do we determine the efficacy of my lead candidate in a complex human tumor microenvironment, such as one that would be mirrored from the actual tumor within the patient? And questions around, of course, the context, how the drug might behave outside the context of the human tumor microenvironment. And then finally, how does this drug interact in a combination therapy, which I'll be addressing as well. So we at DiscoverX have developed technologies to address a number of these questions, and that's what I'm going to spend the rest of my webinar discussing, how we can enable you to address each of these questions in a very effective manner. So a quick intro into DiscoverX. The platform technologies that we have at DiscoverX have been effectively used to create both target-based and phenotypic assays. And long known for our target-based assay portfolio, we have continued to develop these assays outside of now what we were initially developed for GPCRs, now to kinases, interleukins, and as I'm going to talk to you about today, we've expanded that portfolio to develop assays for the checkpoint receptors, enabling cancer immunotherapy development. Since 2012, as we acquired a company known as BioSeq, DiscoverX also expanded their portfolio of assays and services to include a primary human cell profiling assay technology known as BioMap that applies more of a target agnostic approach and provides a phenotypic systems biology tool for drug discovery. And so using these technologies that we've developed at DiscoverX, we can actually enable the development of your cancer immunotherapy molecules at multiple stages in this development process. So if you look at screening and lead optimization, I'm going to talk to you briefly about how our pathfinder checkpoint assays can enable that. And then the same assays that can enable your screening and lead optimization for your drug candidates can also be utilized for lot release applications of your biologic molecule as you get further downstream in your development process. And then I'm also going to touch upon some of the biomap technologies, namely the oncology systems, driving a better understanding of efficacy and biomarker selection, the diversity plus panel that enables us to understand how the drug might behave outside the context of the human tumor microenvironment, and the biomap combo elect strategy to address or assess the question of how these assays might affect clinical combinations and drive your strategy for developing a clinical combination before moving it into patients. So to illustrate this point or this process, I'll be presenting a retrospective case study with pembrolizumab, an anti-PD-1 drug from Merck, and its applications through the various stages in this process. So the first question here, as we look in the first stage of drug discovery, is how do I identify a functionally active lead molecule that then we can take forward? And it seeks to address this to create the appropriate lead molecules taking forward. In this sort of a screening lead optimization assay, there's a strong need for a functional cell-based assay that can enable you to identify your lead molecule and therefore optimize its functional performance in these assays. And this poses a number of challenges during assay development. Namely, is that there is the need for specific cell-based assays for each of these targets, which oftentimes each assay can take 4 to 6 months to develop before actually being able to utilize it for a screening application. These assays often require human cells with complex TCR signaling, which often leads to the question about donor variability, especially if you're using these human cells right out of a person. And these assays oftentimes end up with long assay times and complicated protocols, which reduces or increases the timelines and reduces the throughput of each of these assays. So here at DiscoverX, we sought to address these specific challenges so that we can create a system that allows you to address this question in a very simple yet effective manner. And to do so, we utilized our enzyme fragment complementation technology. So I'm going to just briefly introduce to you our EFC system, or enzyme fragment complementation as it is. It's at its heart, it's a split enzyme system where we've taken beta-galactosidase and split it into 2 components, enzyme donor and enzyme acceptor. When they're apart, they are completely inactive. However, when they come together to create an active beta-gal enzyme, this can then hydrolyze substrate to give us a chemiluminescent output. Using this very simple technology platform, we were able to create an assay. As I mentioned, as an example, I'm going to talk to you about the PD-1. So we were able to create a very effective assay for the PD-1 receptor based on the known biology of the PD-1 receptor. So here what we are showing you is a schematic of how the assay is designed. In the immune cell background, we've expressed the PD-1 receptor tagged with this small enzyme fragment called the enzyme donor. We also have expressed the SHIP protein that is tagged with the EA domain, the larger fragment of our EFC system. Based on the known biology of this receptor, when the receptor gets activated by either PDL1 or PDL2, it leads to the phosphorylation on the intracellular surface of that receptor on the ITIM or ITSM domains by Sarc family kinases that are known to phosphorylate the PD-1 receptor. This phosphorylation event then leads to the recruitment of the SHIP phosphatase to this PD-1 receptor, and this SHIP phosphatase is critical in attenuating any TCR signaling response that is observed in the immune cell. What we've done here by tagging the SHP protein with the EA tag and the PD-1 receptor with the ED tag, we've enabled us to identify the activation of the PD-1 receptor through this EFC system. So once PD-1 gets activated, we get the SHP recruitment, brings EA and ED into proximity, forces them to complement, which then creates an active enzyme hydrolyzing substrate and giving us light. So in the presence of chemiluminescent light, we know that now the PD-1 receptor is active, and as we see a decrease in light production, we know that we are disrupting the interaction between the PD-1 receptor and, the activation essentially of the PD-1 receptor. So using this simple approach now, where it mimics the biology of the receptor, where the PD-L1 or PD-L2 is displayed by the cancer cells, and the PD-1 is being displayed by the immune cell background, it allows us to create a very sensitive and very easy to use screening assay to enable you to identify either inhibitors or activators of the PD-1 receptor. And so here, what we are looking at is an example of where we've tested pembrolizumab, the anti-PD-1 drug from Merck, and nivolumab, which is the anti-PD-1 drug from BMS, in our PD-1 assay. And what we can observe here is, as we increase the concentration of these two drugs, we see a decrease in the amount of light observed in our assay, again indicating that we can see an inhibition of that signal or the PD-1 receptor activation using this approach. And what's also striking is that we can observe this assay at single-digit nanogram per mil sensitivity, which is at least 15 times or greater more sensitive than any of the other assays that are out there for the PD-1 receptor. So this again drives the idea that we can use this approach to identify biologics that might inhibit the PD-1 receptor. And strikingly, what we can also observe is that we can also identify small molecule inhibitors of the PD-1 receptor using this assay. So on this approach, what we are looking at is two different small molecule kinase inhibitors, dasatinib as well as XL2880, both of which are pan-SRC family kinase inhibitors. And as we increase the concentration of these drugs in our system, we can see a dose-dependent decrease in the amount of signaling that is observed in our assay. That being said, this indicates that we can actually utilize this to screen for either the kinases or the kinase inhibitors that might be specific for PD-1, as well as disruptors of the receptor ligand interaction, again, through either biologic or small molecule approaches, giving us a very simple, easy to use screening assay using this technology. So in summary, what I'd like to indicate in here is that we've developed, using our Path Hunter checkpoint assays, a technology platform that enables screening and lead optimization for both co-stimulatory receptors such as OX40, CD40, and HVEM, as well as co-inhibitory agents such as PD-1, VISTA, and TIM3 using our assays. And these assays all actually have a very simple, easy to use protocol with results in less than 5 hours, again enabling these assays to be utilized very effectively in screening and lead optimization campaigns to give you data responses very quickly in these approaches. These assays, as I indicated, can support the development of biologics and small molecules and enabling you to screen with confidence because of the sensitivity of the response that we've observed in these assays. And the same assays that we've developed here can also be applicable for QC lot release testing of biologic drugs as these molecules move further down in the development campaign for you. So once you've identified a lead molecule that you know that is and then optimized it. A question that often comes up is, how do I test the efficacy of this molecule in a more physiologically relevant system? And in particular, in the case of cancer immunotherapies, that the question revolves around, how do I test this molecule in a complex human tumor microenvironment which mirrors what would be observed in an actual patient tumor? The challenges associated with trying to address that question is that, in vitro assays that have been currently utilized do not actually recapitulate the complexity of the human TME, because oftentimes these are monocultures of these assays. What we also observe is that animal xenograft models, which have been very, very valuable for a wide variety of approaches, are not really amenable for high throughput or even medium throughput profiling of compounds to determine the efficacy of these small molecular biologic drugs. And finally, the lack of appropriate response biomarkers hinders the translational interpretation of these drug responses for efficacy and safety of a drug once the molecule goes into the clinic. And so the systems that we've developed here really can enable the addressing of this question and hopefully overcome a number of these challenges that we've seen over here. And so to address this question, we've developed, as I mentioned, the human primary cell-based phenotypic screening platform known as BioMap. This uses primary human cells that are co-cultured and stimulated in such a way so as to mimic or mirror the different aspects of human biology. We use primary human cells that are pooled from multiple healthy donors and in such a way so that they can capture the interactions of these cells within various tissues. And being fully human, these systems also are amenable to identification of biomarkers that could have impact in terms of translational impact on efficacy or safety of the actual drug in a patient environment. So this system allows us to now profile small and large molecules or combinations, and using our bioinformatic and data mining tools that we've developed, it allows us to integrate the phenotypic signature or activity profiles of each of these molecules into our BioMap systems to help us translate this data to create, the appropriate response or to identify the appropriate response, and progress molecules into safe and efficacious drugs. So diving into this a little bit deeper, in particular, when we start thinking about the BioMap human tumor microenvironment models, to develop this, we've developed two systems in which we use primary human PBMCs co-cultured with either primary human fibroblasts or primary human epithelial cells that are from the vasculature, the HUVEC cells, alongside a cancer cell line that can then mimic either a stromal or a vascular tumor microenvironment. In this case, I'm going to show you data with the HT29 human colorectal adenocarcinoma cell line. However, we've also developed a similar model with the non-small cell lung cancer cell line and are developing further models for other types of tumors and tumor microenvironments. So in this case, we've optimized the cell culture of the fibroblast and the cancer cell lines and stimulated the immune cells through the T cell receptor to create an inflamed tumor microenvironment, as would be seen in a real human tumor. And these systems are now known as the BioMap oncology systems and are available to test agents, either small molecular biologics or combinations, as I indicated earlier. And then we can then monitor the impact of these by looking at clinically relevant biomarkers, protein biomarkers that are being modulated in each of these systems. As with the development of each of these systems, an important question that always comes up is, how have we validated this system? So here, what we are looking at here is some validation data that clearly is able to show that this system is able to mirror what's observed in a tumor-mediated immune suppression microenvironment. So in the graph here, we are looking at on the y-axis the relative expression levels of interferon gamma. On the x-axis here, we are looking at various different conditions or co-culture systems or monocultures that we've utilized to test this. And in the green is the unstimulated condition. Again, as I mentioned, we stimulate these systems with via the T cell receptor to create an inflamed environment. And in the orange, we are looking at the stimulated system as to recreate this inflamed environment. As we would expect that with PBMCs, as we stimulate them through the T cell receptor, we can actually get an increase in interferon gamma. And when these cells are co-cultured now with the fibroblasts, we get even a further increase in the production of interferon gamma, which again is something that we expect to see, and it's typical for something like an inflammation response, for example, as observed in rheumatoid arthritis. However, what's striking is that once we add the cancer cell line, the HT29 cell line, to either the co-culture of fibroblasts with PBMCs or the HT29 cell lines to just the PBMCs alone, we can see a strong suppression of this interferon gamma response. Again, what that indicates is that we can see that this system is now able to mirror the immune suppressive microenvironment of a real human tumor, as would be observed within a patient. And so that's actually something that was that's important, because I think we'll come back to this point as we go further into the webinar. A question that often comes up around this is that we are using primary human cells to develop an assay, and so therefore there is the potential for donor variability. So another aspect of the validation that I wanted to demonstrate here is that we wanted to create a system where we can minimize this donor variability by providing a platform that can be utilized for screening and profiling in a much more reproducible manner with very consistent results. So to test that, we tested our assay again with either PBMCs that have been pooled from multiple donors or PBMCs that are derived from single donors in this case. So as you can see within the two conditions, here again we are looking at the non-stimulated, which is in blue and pointed out by the little blue arrow here, and the stimulated in the dark red or the lighter red colors. And again on the y-axis we are looking at the interferon gamma response in these assays. What's immediately apparent with this graph is the robustness and the reproducibility of the system, with PBMCs that have been pooled from multiple donors is much higher than what we can observe with the systems that use single individual donor PBMCs. So this is one of the major reasons why we decided to work with pooled PBMCs to increase the reproducibility of our system and minimize the contribution of individual donors in this system. However, when you pool PBMCs from multiple donors, immunologists ask the question about MLR biology, and indeed that is a very, very valid question. So if you take a look at the non-stimulated conditions in this assay, where the PBMCs from multiple donors are now in a well together in all of these cases, where we can see very little activation going on here in these assays at 48 hours as the endpoint or the duration of our assay, compared to the controls, which are all of the individual donors. It indicates that there is very little MLR actually going on in our assay, and the response that we see in our assay is really coming in from the stimulation and activation of these T cells in our system. So these are the systems that we've now developed that we use to screen and profile drug candidates and to look for their impact on protein-based, clinically relevant biomarkers. And the end result of a profile using the biomap system looks something like this. So here we are looking at the STRO HT29, which is the stromal HT29 system that is a biomap oncology system, as I mentioned earlier. Along the x-axis, we mark out the various different protein biomarkers that we are reading out in the system, either directly, again, in the co-culture or as soluble forms of that protein in that supernatant. On the y-axis, the data is shown as the relative expression of that biomarker when treated with a particular concentration of the drug, and here we have tested 4 different concentrations of that drug, compared to the vehicle control of that drug on that same plate. And the gray envelope here, as we observe, is the 99% confidence interval of the historical vehicle control data for that particular readout in this particular assay. So it allows you to see how your drug affects any particular biomarker in comparison to all of the historical controls that have been run, or another way to put it is in comparison to all of the historical noise of that particular biomarker activity using this profile. So here, just as an example, we've shown the GSK's MEK inhibitor trametinib profiled in this system. And as you can see, it has a number of different activities, and I won't really go into some details, but a couple of things that I just do want to point out is it is actually showing a further suppression of the immune response in this human tumor microenvironment model, as observed by a decrease in a variety of different cytokines observed in this assay. So using this same panel for the stromal tumor microenvironment as well as the vascular tumor microenvironment, we tested pembrolizumab in this assay, which is, as I mentioned, the case study that we will be walking through. As we can see here, we are looking at 4 different concentrations of pembrolizumab and run through on the stromal and the vascular HT29 systems. And what's striking here is that we can actually see increased cytokine production across both the stromal and the vascular systems. As you recall from a couple of slides previously, I'd shown you that the immune function of these systems was actually being repressed by the presence of cancer cells in the system. So therefore, this activity, what we observe here as increase in the amount of cytokines being expressed, is actually largely represents a restoration or recovery of that immune function in a human tumor microenvironment in the presence of pembrolizumab. And this is really exciting because this was exactly what we would expect to see based on the mechanism of action of this drug, where it is alleviating or recovering this immune function that has been suppressed by the presence of a human tumor. In addition, because all of the cells inside the system are human cells, we also have the potential to identify response biomarkers, or predictive response biomarkers that may have a translational impact once these drugs move into the clinic to identify patients that might be responding appropriately to these drug molecules. So we've taken this approach with pembrolizumab and we've applied the same assay to nivolumab, which is again an anti-PD-1 drug from BMS. And what we can observe here is that both of these assays have strong overlapping functions, as would be expected between the two. So this approach actually highlights the idea that it can enable you to benchmark your drug candidates against existing molecules, and it can also be utilized to confirm the target class signature biomarkers of efficacy for your candidate molecule before you move this forward. So again, allows you to benchmark, do some lead optimization, and as well as identify signature biomarkers of efficacy before moving this molecule further along into the clinic. And of course, the goal of an approach such as this, or the panel such as this, is to actually enable you to test new candidate molecules. So here we have two different molecules that have been overlaid. In green, we are looking at paclitaxel, and in red, we are looking at anti-CD73, an antibody that was tested in a collaborative study between MedImmune, AstraZeneca, and DiscoverX. So let's focus on the red line initially, which is the anti-CD73 antibody from MedImmune, AstraZeneca. Again, in this case, what we see here is an increase in cytokine activity both in the stromal and in the vascular system, indicating, again, a restoration of the immune function or immune activity in these systems in the presence of the drug molecule. Interestingly, although the patterns are quite similar between the anti-CD73 and the Pembro drug from the earlier slide, there are certain differences between the two, and I won't really dive into that, but this sort of an approach enables you to tease out differences in terms of functional activity between two different molecule types targeting similar receptors but different specific targets, enabling you to kind of differentiate your molecule compared to other molecules. Here we've also included the clinical drug paclitaxel. This was really surprising data, but it showed a pattern of immune recovery in both the stromal and the vascular systems that we can now confirm is actually a mechanistic signature of what we observe in as we've observed a similar pattern from a molecule known as epithylamine D, which is also a microtubule stabilizer, and therefore this effect that we observe here is actually more is representative of that mechanism of action in this particular system. So we've seen similar data from other clinical drugs, again indicating that these clinical drugs that can restore immune function in this immune-suppressed human tumor microenvironment will give signatures of this increased cytokine activity in these systems. So this approach enables you to profile your drug candidates in an oncology in our biomap oncology systems, and can enable you to screen for and identify molecules with the best functional activity to then take forward as your lead molecules in your program. So now that we've identified a lead candidate and determined its efficacy in a human tumor microenvironment, a question that comes up is, how does this molecule perform or interact with the tissues outside the context of this human tumor microenvironment? Our senior director of R&D often says that we are making these drugs to treat our patients and not just the disease. And so a question like this is a really critical question, because it allows us to now look at how might this drug affect the patient as a whole. And so one application of our biomap system that we've developed has been to test the drug molecules in an environment where we are looking at microenvironments outside the context of the human tumor. And so when using current approaches, there are several challenges that our clients have faced when trying to address this question. And that's largely because there is a reliance on single cells or organs that and then as well as a bias towards specific targets and target pathways to address this question. These assays often are much less complex and capture isolated biology, and there's limited testing of this in a broad-scale human biology system with a drug candidate before it actually moves into this clinic. So therefore, to address this question, the biomap phenotypic platform takes a systems biology approach to capture broad human biology in a target-agnostic manner. And the outcome is what we observe here is the outcome from our Diversity Plus panel, where we have 12 different primary cell co-culture systems that are all based on normal human primary cells that are co-cultured to model different disease processes, and none of these co-culture systems utilize any cancer cells or cancer cell lines, again, indicating that each of these is more reflective of what we would observe in a normal biology outside of the human tumor microenvironment. So we have a broad range of tissue types that we can model here, ranging from vascular endothelial cell inflammation to vascular smooth muscle inflammation, immune cells that are mimicking various different activation states in either monocultures or co-cultures, to systems that can model epithelial inflammation, matrix remodeling, and fibrosis, as well as wound healing type approaches for using these co-culture assays. So in this case, what we've done is we've profiled erlotinib at 2 different concentrations. And as you can observe, we can largely see an activity in this bronchial epithelial system for erlotinib, which has been this system has been stimulated with some inflammatory cytokines, creating a bronchial inflammation model. And this is exactly what we would expect to see based on erlotinib's primary indication of oncology. So in addition to this effect on matrix remodeling and epithelial biology that we see here, we also see a signature activity of EGFR kinase inhibitors, and that's an upregulation of EGFR in the system itself. And this is actually something that we've seen with multiple EGFR kinase inhibitors that are in our database of over 4,800 compounds that have already been tested in the system and are part of the database that we've got these that we can test against. This signature activity of EGFR is indicative of a skin rash, and this is something that is now clinically monitored in patients, as the presence of a skin rash in patients that have received erlotinib is indicative of a patient actually responding to the drug. So this sort of an approach allows you to evaluate the impact of the drug in the human biology, in broad human biology, outside the context of the human tumor microenvironment, enabling you to either identify a mechanism of action if that is unknown, or identify biomarkers of efficacy, as we discussed in this particular instance. Another example that I want to touch upon briefly is paclitaxel, or Taxol, as we talked about. And this is, again, something that we looked at in the Diversity Plus panel in the absence of the immune suppressive cancer cells. What's striking here is that in the absence of the immune suppressive environment, we don't actually see any immune activation at all with paclitaxel, with using our cytokine, monitoring the cytokine production in these assays. As we had observed, something similar with the human tumor microenvironment, where the use of paclitaxel in an immune-suppressed human tumor microenvironment led to an increase in the amount of cytokines, indicating a reprieve in that immune function. So outside the context of the human TME, paclitaxel actually showed a inhibition of proliferation for either vascular epithelial cells or vascular smooth muscle cells, both of which are indicative of or associated with a cardiovascular adverse event. However, this was something that was not observed in the human TME, again, allowing one to position that drug for a particular indication, in this case, of course, for oncology systems. So one of the most exciting profiles that we observed in this was actually for pembrolizumab, as continuing along with our case study, and this really showed that at a concentration that was active in the human tumor microenvironment from a couple of slides ago, we see no activity of pembro across our human cell system outside of the tumor microenvironment. the lack or the inactivity in this case where in systems that have primary human cells without cancer cells is consistent with the mechanism of action of this anti-PD-1 drug, which indicates that it is able to restore immune responses in an immune-suppressed environment without being inherently inflammatory. And that was actually something that was, again, it's what's known quite well about this drug, and it's something that we've observed in this approach as well. We've also tested nivolumab, which is Opdivo, and we've seen a very similar profile across the board. Again, having a very consistent performance with what's known about these drugs is that they're able to restore immune responses in the tumor microenvironment, but are not inherently inflammatory. So again, this allows you to confirm the selectivity of your molecule, both on target or even target-related secondary effects, and allows you to identify or inform the mechanism of action in these approaches. So at this point now, you've identified, screened for, identified a molecule that is safe and efficacious. A question that the cancer immunotherapy field as a whole is hurtling towards at a very, very high speed is, how does my drug perform in combination with drug X or with other molecules that might already be in the clinic or are being developed at the same time? So, and the question that is how does this perform in the context of combination? And this could be either a rationally designed combination therapy, or as in many cases, how does the molecule interact with the standard of care drug that might already be in the patients inside the clinic? So the challenges with regard to testing these clinical combination strategies prior to actually the patient is quite challenging. It's technically complex to test these combination therapies. Xenograft models that exist have, again, been very valuable for a variety of studies, but are often poorly predictive of how these combinations may perform in humans, and tend to be quite expensive, especially when looking at broader applications of these for a variety of different combinatorial approaches. So that leads us to a point where these combinations are often tested directly in patients, which is not an ideal environment. So to address this question, to address this challenge, we developed a ComboElect platform, again using our biomap systems, and we've developed a strategy to test these molecules in combination, where both molecules are tested alone at increasing concentrations in a 4x4 matrix. So this is all done in a single experiment, so we can rigorously test whether the combination that you want to test is either different in terms of safety and efficacy than either agent alone. And the value of this response or the assay is really twofold. One of them is that it gives you a clearer in vitro answer on whether the combination that you intend to put into a patient will be safe and efficacious compared to any of those monotherapies. And the second is it allows you to address what is the sweet spot for your combination before putting it into the patient. Is it concentration X versus or Y of the two before taking it into the patient system? So as an example of what this output gives you, we are going to look at just one of these slots in this matrix. So just one of these over here, highlighted in this graph. What we can observe here is in blue, we are looking at paclitaxel. In red here, we are looking at pembrolizumab, and in black, we are looking at the combination of pembro and Taxol at the same concentrations as tested in the monotherapies. And what's striking here, as indicated by these pink arrows, is that in a number of cases, we can actually see, we can observe a effect when the molecules are being treated in combination, and this additive effect can be observed in the increase in cytokine production in either the stromal or the vascular systems in a number of these cases. Again, indicating that the presence of these molecules in a combination might actually have a positive impact on compared to each of these in monotherapies. So with that, I'd like to now summarize how the DiscoverX technologies have now been can enable and have enabled our clients to develop cancer immunotherapies all the way from screening to clinical combinations. Our Pathhunter assays have enabled our clients to do screening and lead optimization to identify the best lead molecule to take forward. Our biomap oncology systems enable the determination of drug efficacy in a human tumor microenvironment, including the identification of efficacy biomarkers to take forward into a translational study. The Diversity Plus panel profiling has enabled the testing of these drugs in a normal tissue system that is outside of the context of the human tumor microenvironment. And finally, the combo-like systems enable the testing of these drug combinations in an in vitro setting, so you can determine the best combination to take forward as well as the best concentration to take forward for that combination into your patient populations. Another application that we've recently worked with a few clients on is before in-licensing molecules, especially in the cancer immunotherapy space, there is discussion about how these molecules might interact or test with, work with other existing drugs in a combination setting. And so this system allows us to give our clients some predictive info of how these molecules might interact in a combination setting before they decide to move forward with with partnering with another client. So overall, the DiscoverX platforms have enabled us or smoothed the process of cancer immunotherapy development for a number of our clients, and can do the same for a variety of your systems. So with that, I'd like to end this webinar. Encourage you to find out more about our different platforms and technologies as we spoke about at our website, which is discoverx.com. And if you do have any questions that I am not able to get to at the end of this webinar, please shoot them into the email addresses shown there, and we'd be happy to address those. So with that said, I'd like to thank you for your time and your attention. Again, as a reminder, any questions that you have, please type those into the chat box or the Q&A box that you can find in your dropdown from your WebEx, and I'd be happy to address them as they come in. So the first question that's come in here already is that how long do we run our systems for? So the Div Plus system that we've talked about, which is our broad panel human biology system, is typically run for about 24 hours. Our oncology system, which is the biomap oncology system, recreating the human tumor microenvironment, is run for approximately 48 hours, and we also have other systems like the Myo5 system, etc., that is run for 48 hours. So those are our standard run times that we run these assays for. So a question that's come in is, do we have a pooled PBMC MLR phenotypic profile data at 48 hours post-TCR activation? So all of the data actually I showed you with regard to that human tumor microenvironment system is actually at 48 hours post-TCR activation. So in all of those cases, we are looking at data post 48 hours after the activation event. So another question that's come in is, what's the average number of PBMCs, PBMC pool donors that we utilize per experiment, and what is the percent CV between these assays? So that's a very good question. as I showed you, as I indicated earlier, the um, the the percent CV between each in this particular experiment was about 11% for using our pooled PBMCs, and this was looking at 11% between multiple different experiments using different pools of of PBMC donors. In terms of the average number of donors we utilize, it can range anywhere from 3 to 5 donors in each pool of PBMCs. Now, the question that's come in is, what's the ratio of PBMCs to cancer cells in our vascular system for biomap? That's a good question. I do not have a specific response to that answer just yet, but we can definitely follow up with you directly to get you a more specific answer, because I do not want to misspeak on behalf of that ratio. Now, the question is that we've got, do we have other cancer cell types that we've developed these biomap oncology systems for? Excellent question. As I mentioned, we've developed this for the HT29, which is the colorectal cancer cell. We've also now developed a similar approach for the using a non-small cell lung cancer cell line. So that is the second one that we've got available, again, in our biomap oncology systems. And we are actively developing these approaches for other types of cancer cell types. And if there is any particular one that you have in mind, we'd be interested in hearing about it. And there is definitely a custom process where we can where we may be able to custom create a system for your application as well, for your particular cancer cell type. Now, the question that's come in is, does a particular drug molecule, sorafenib, also enable recovery of this immune response? So that's a good question. It is something that we would have to look into our database, and we can provide some feedback to you directly with regard to that question. Another question that's come in is regarding the screening assay that we've developed in the past. Are each of these assays developed for a co-culture setting? That's a good question. With regard to the PD-1 assay, as I indicated, it is developed as a co-culture where we've got the ligand, the PD-L1 or PD-L2 being displayed on a cancer cell line, and the PD-1 receptor being displayed on a immune cell background. Apart from that, we've done most of the other assays that we talked about, for example, CD40 and OX40, etc., are using soluble ligand. We have not tested it with a co-culture approach, but I anticipate that with the use of soluble ligand, this enables us to move the assay relatively quickly, although the use of a co-culture system will also or may also give us similar data in using for those assays. So another question that's come in is, how many combinations have we tested using our ComboElect platform? That's an excellent question. We've actually done a number of different combinations where we've done biologic combinations, biologic small molecule combinations, small molecule combinations as well, and we've tested it using that the ComboElect platform that we described earlier. And that's a number of that has actually been done, or a lot of that has been done for client purpose that we, due to confidentiality, cannot share that data. However, we do have additional data for combinations that we've done that is shareable, and it is something that we can definitely provide to you on request with additional case studies. So yeah, the the question that came in is that it seems that we've made an observation that paclitaxel may have an immune stimulatory effect. Is that correct? I just want to clarify that what we've observed in this system is that actually the paclitaxel has it alleviates immune suppression. So it's not actually having an immune stimulatory effect as observed in our Diversity Plus panel. That's an important consideration outside the context of the human tumor microenvironment as observed in our Diversity Plus panel, where there's no tumor cells at all. Paclitaxel does not have or does not stimulate the immune system. Conversely, in a human tumor microenvironment where where the immune system is actually being suppressed by the presence of the cancer cells, only in that system the presence of paclitaxel increases immune function, and therefore essentially is restoring immune activation in an immune suppressive environment. So it's a fine distinction, but it's still fair to say that paclitaxel does not have an immune stimulatory effect on a broad level. So all right, so since we've come to the end of our time, I'd like to again thank you all for your time, for signing up, and for attending this webinar, and for the engagement that we've observed here with the number of questions that come in. Once again, if you do have additional questions with regard to this webinar, please send your questions into those 2 email addresses that we've written up on the slide here. Additional data and the support for all of the assays that we've developed in this presentation or presented here are available on the discoverx.com website. And as a reminder, this webinar is being recorded, so within the next couple of days, you will have a copy of the recorded webinar as well as a copy of the slides to look over again or to distribute amongst your colleagues. So thank you again for your time, and we look forward to continuing this discussion with you and on a more personal level, and as well as working with you in the future. Thank you again, and have a fantastic day.